Okay, well, thank you very much for the opportunity to be here. Although I'd rather we were all together, I'm sure that's the common sentiment. And actually right where I'm sitting in Cambridge, Massachusetts, we're having a thunderstorm right now. So that if my power goes out, that will be the explanation, and you won't have to worry about that. Okay, so let me tell you a little bit about our lab in general. What we've been interested in is bringing computational and systems approaches to bear on complex pathologies and how to treat them. So what's shown here is a tumor microenvironment on the left-hand side, upper. In the middle is women's reproductive disease known as endometriosis. Below that, diabetes, and then down in the bottom left-hand corner, inflammatory bowel diseases. And these are all just examples of complex pathologies that are poorly understood due to the myriad molecular and cellular interactions and very difficult to treat. We tend to work these days mostly with in vivo experimental systems, so mice, shown on the right, humans shown on the right and the bottom, primates also. And what we're after is that we start by interrogating at a molecular and cellular level animal models and human subjects, and from those experimental data, try to understand what's going on in these complex diseases. What I want to focus on today is if you do that, let's say you do studies in mice or you do studies in non-human primates, ultimately, of course, we're interested in human disease. So how do we more effectively translate whatever we find from animal studies to a human? And this is the huge gulf there is between preclinical to clinical translation in the pharmaceutical industry, which is a very, very big problem these days. Now, it shouldn't be a surprise that this is a problem because there's numerous reasons why relationships between molecular data and phenotypic data would be different for humans compared to rodents or primates. So how do people do it now? Well, what people do now is look for commonalities. Can you observe commonalities in the genes, in the proteins, in the cell types that are expressed or active in your mouse experiments? And I'll just talk about mouse today, or your human experiments that you think are related to pathophysiology and treatment. So if you find some, let's say, differentially expressed genes in mice related to some disease, and there's some, they are in common with some differentially expressed genes in humans with that disease, you say, okay, that's what translates. All right, it should go without saying that this approach has some value, but it has severe limitations. Because if they did not have severe limitations, then there'd be much greater success in translating from preclinical to clinical in the pharmaceutical pipeline. And we know that success rate is typically only about 10%. So we do need to do better. So what I'm going to do in my presentation, first, just give you a conceptual basis for our approach, which is a computational systems approach. Otherwise, we wouldn't be here in this meeting. And then I'm going to give you two examples out of the number that we've done so far. So first, to understand what we're after. So let me lay this out schematically, that what people try to do these days, if you see in the upper right-hand side, is they take their animal experiments and they get molecular or cellular data, transcriptomic, proteomic, things like that. And they try to relate that to the phenotypic data of disease versus normal or response to treatment versus not. And then what they look for is, are there any commonalities if they have any human data also related to that phenotype? Okay, and that's what's works marginally well and needs to be improved. So our approach looks something like this. Instead of translating observations, what we're going to do is translate computational systems models that in the animal, you start with constructing a computational model that relates the animal molecular cellular data to the phenotype. This model can be of different types. We'll talk about that in a minute. And then what you do is you humanize that model. People talk a lot about humanized animal models where they try to make them more like humans. Well, we're trying to make the computational model more like human. And one does that by, even if one has only limited human data, you use that to essentially update the parameters in the computational model. So that's really the notion. If you want to read more about this notion, we wrote a Science Perspectives earlier this year. Okay, the biological premises for this I can outline here. These are really important to understand. So the first is that any particular molecule doesn't necessarily translate across species. So if you find some gene that seems to be partially responsible for a disease in mouse, it doesn't mean that same gene or protein is going to be have that same effect in human. However, we do believe that explanatory mechanisms translate, otherwise we wouldn't really believe in biology as unified across species, especially mammalian. Okay, so we're looking for explanatory mechanisms rather than particular molecules. Now, you also have to appreciate that there's generally not a single explanatory mechanism for a disease or a responsiveness to treatment. There's likely multiple of them, and they're all going to have some quantitative contribution. And those quantitative contributions might be different in mouse, say, than in humans. That if there's 3 different mechanisms that all contribute some, maybe in mouse they all contribute equally, maybe in human one of them really is dominant to 90% and the other 2 only contribute a few percent each. And that is entirely reasonable physiologically. So what we need to do is infer these explanatory methods from the computational models of molecular pathways and processes in how they predict phenotypic data. We first do that in the animals, and then we translate to the human or to a different animal species by quantitative modulation of those model parameters that relate the molecular cellular data to phenotypic data. So those are the premises. If you have some issue with those premises, then the actual approaches, won't satisfy you. What kind of models are we talking about? Well, first of all, what kind of data are we talking about? Well, in the end, up in the right-hand corner, we want to be able to predict or interpret or explain physiology and pathology and treatment response. And so there's got to be some kind of characterization of that phenotype. It might be at an organism level, it might be at a cell or tissue level. We want to explain it in terms of essentially molecular processes. Might be transcriptomic, might be proteomic, might be post-translational. So we want to build models that take the data in the blue box here and use it to explain the data in the purple box up there. That's the kind of data we'll generally be after. What kind of modeling approaches? Of course, you can use anything that you think is going to be beneficial. What I will say is that for most of the problems we've been working on these days with these very complex diseases and in vivo data, it's very difficult to write theory-driven differential equation, physical chemical models because we just don't know all the key variables or their interactions. So we've tended to live on the data-driven side with machine learning methods of various types. But the translation can be done by theory-driven models just as well if you have the data that bear it. All right, I want to make one point about a crucial part of these machine learning methods that differ from traditional multivariate analysis that is done more in the statistics field. Traditional multivariate analyses generally assume that input variables are independent, that there's really no co or anti-correlations among them. If that's not true, then those methods really break down. Of course, that's a terrible assumption for biology, because in biology, with all its complexity, there's a tremendous amount of co and anti-correlation among variables. So you need methods, and machine learning methods certainly satisfy this, that accommodate covariation among input variables, and in fact, that it's an important feature. So what this means is that what we're going to be doing is developing latent variables, that can also be viewed as dimensionality reduction, where you take all the variables you have and you reduce them to a number of combinations of variables, because those combinations of variables are co-correlated or anti-correlated. And so they provide a latent variable that has the actual biological process information. All right, so what does this look like? Well, on the left-hand side is the way people in biology typically think about, or there might be a particular variable, a transcript level, or a protein level or activity, and that might vary with the different subjects or treatments, and then they might want to try to relate that variable to the phenotypic response, like a tumor response. And typically, this is very difficult to do because there's rarely univariate relationships and monotonic relationships between any individual variable and any kind of dependent phenotypic response. So what latent variables tend to do is then to invert this mathematical problem and look in the space of all the variables. So what's shown in the middle is all the variables involved, might be 20 or 100 or 1,000 or 10,000. And then each of these points is the different subjects and treatments, and they're colored by what their phenotypic response is. And what you're looking for is the data structure and the latent variables that explain then different levels of phenotypic response. So they might be as simple as principal components shown on the right, but in any event, what you want to do is constructing combinations of individual variables that do exhibit substantive covariation. And these latent variables are where the biological information is, and that's what's going to be translated. Okay, so that's the philosophy, that's the premises, that's the conceptual basis. It may be attractive to you, it may be anathema to you. Nonetheless, that's our reasoning. So given that, there's 3 papers that you could read. 2 are published already, and 1 will be out in a month that gives specific examples of doing this kind of thing in very different kinds of problems. So one problem is relatively easy where the data types are similar between your animals and your humans. One type at the bottom is very difficult where there's entirely different data types. And then there's kind of a middle type in which the data are moderately different. And each of these really needs a different computational framework. So I'm going to talk about the first 2. I'll leave the other one for you could look up if you want. It's really kind of a meta-analysis with different types of statistics. The first 2 really fall into systems modeling much more closely. All right, so I'll give you these 2 examples and then we'll be done. All right, so the first one was what we considered a really basic place to start. And this was, you can find in the literature and in some reviews, a number of case studies of inflammatory pathologies where there's matching transcriptomic and phenotypic data sets between mouse and human. So on the right-hand side, this is a subset of the 36, and you can go into the databases and find them. There might be sepsis, burns, endotoxemia, trauma, different mouse models for each. And then from blood or tissue samples of various types, there's transcriptomic data from the humans and transcriptomic data from the mice. And they have this for control healthy individuals, and they have this for diseased individuals. Okay, so this is a really, really good set of data with multiple case studies. It's also a good place to start because in the last 5+ years, there's been tremendous disagreement about how best to analyze these data in the literature. So I've shown down below a number of references, and there's many others. And what's the bone of contention? Well, shown above here are some plots of, if you have for the different case studies, mouse gene expression on one axis and human gene expression on another axis, and each one of these little squares is a different one of the case studies. And the argument is about, well, how much are some of these transcriptomic data in common? Again, people looking for the commonalities. So basically looking for what's correlated, what would be on the 45-degree lines between mouse gene expression and human gene expression. And surely those would be what's most explanatory. Okay, so looking for the commonalities. And as I said, if you read all these papers, there's disagreements about how well this works and what you learn and what you don't learn. So we thought this was a good place for us to bring in our new computational translation method shown schematically here. So we'll start with any of the case studies with a mouse disease model, and Y sub M here would be the molecular data for a mouse. X sub M here would be the phenotypic data for the mouse. These are mostly classifications between disease versus healthy and transcriptomic data. And again, typically what's happened is to look for commonalities between those relationships and what would be in the human patient cohort on the right. What we're doing instead, again, is we're going to construct a machine learning model based on the mouse data, and then in either supervised or semi-supervised fashion, apply that to the human data and figure out what the weighting coefficients are that change the predictions of the mouse data so that they're useful for the human data. We use many different types of methods in that paper. What I'm going to talk about here just briefly is artificial neural network, just because of the great interest in things like this these days. Of course, you're well familiar with this, perhaps, that it's used very heavily in things like image analysis, where you're going to take image pixels and decide whether an image is more likely to be a cat or a dog. So what goes in your input are your image pixels, they're your predictor variable measurements. You construct latent variables, combinations of those pixels, and then find the right weighting coefficients that take those latent variables and do your classification for cat versus dog. So latent variable construction and then parameter weightings for the contribution to the result. Well, we're going to do the same thing, but now our predictive variables, instead of imaging pixels, they're molecular omics like transcriptomics. And instead of predicting cat versus dog image, we're going to try to classify disease versus healthy patients or mice. But again, it's going to take the transcriptomic data, construct latent variables, and then develop the weighting coefficients for the prediction of the outcome. And what we're going to do for translation is first we're going to construct this on the mouse data and construct the mouse model with the mouse parameters. And then we're going to modulate the latent variable parameters against the human data and see how the weighting coefficients change between what actually accounted for mouse physiology versus what accounts for human physiology. So I hope you get the idea. I won't go into too much detail, but what we're really after is if we can construct models that more faithfully predict human outcomes than they would directly from just the observables of the mouse data. We'll do this in terms of some typical machine learning statistics, precision and recall, basically true positives relative to false positives and true positives relative to false negatives. You want these to be as large as possible, but then what we're going to do when we construct these models is we're really going to be looking for what are the biological features, latent variables that are the key predictors, and what does that tell us about the biology and what's different in human than in the mouse. Okay, we do a lot of regularization so that these models are not overfit. So in any given model, out of a few hundred or 1,000 transcripts, ultimately in any given model, we construct ensemble models. There'll be a few dozen transcripts that form the latent variables. All right, how does this work? Well, I'll just show you the F scores here, and what you want is the largest number to the right is better. So on the bottom, if you just construct the mouse model and try to predict human, it's really not significantly different from 0. Well, it is to a small extent. The different types of machine learning models improve on it. The very best one in this case is a semi-supervised artificial neural network. That doesn't mean that's got to be the case for all studies. But it just shows in general across all 36 case studies that it works so much better than direct translation. If you look across all 36 case studies on this, what you see is, what you want to look for is the difference between the black and yellow bars, and they're much higher than the red and blue bars. And what does this mean? The black and yellow bars are when you've taken the mouse model and humanized it by constructing the neural network model. The red and blue bars are what happens if you just try to predict from the mouse data directly. So in almost every case, the yellow and black bars are higher than the red and blue bars, which means that the neural network modeling has added value to your capability of predicting the human phenotype from the, instead of directly from the mouse data. This improvement can be fairly dramatic. What's shown on this slide is one particular mouse model for sepsis. Well, there's actually 3 different ones over here. And what's shown is from the mouse data themselves, those mouse models, really their differentially expressed transcripts are not predictive at all of what happens in human disease. Yet, if you now run them through the artificial neural network model and the humanization of it, you can predict a very large fraction of the human biology. And so that shows you very, very strongly what can happen. Now, how does this work? Well, this is a really important plot. What's shown here is if you looked at the mouse transcriptomics, on the x-axis are the mouse gene expressions. All these blue dots are different mouse genes and their level of fold change between disease and healthy in mouse. On the y-axis is the same transcripts for human. And what you see by and large is, of course, the differential expression in mice sometimes is the same as human, and that would be the 45-degree line, but mostly not. And most importantly, if you looked for the commonalities, you really would have picked out just a couple genes as common in terms of their differential expression between mouse and human. Whereas what's shown on the right-hand side here is when you construct all these neural network models in ensemble and what fraction of the models that the genes are, any given gene is used in, these are the top ones. And if you look at all these with the colored stars, you see you would never have picked them out from commonalities because they do not have much in common with respect to their expression, differential expression between mouse and humans. They're down in the noise, they're certainly off the 45-degree lines and so forth. So just by sticking to what's in common, you would miss a lot of predictive capability. And consequently, that turns into an increased ability by the computational translation to find pathways and processes that are involved in the human disease that would not be observed by the mouse. So what's shown here in red are, if you go into like gene ontology or gene set enrichment, pathways and processes that you would indicate from the mouse models. In black is what's true in human, is blue is what you get by translating the mouse data through the neural network models. And there's well over 100 that are absolutely true in human that you would not find directly by observation from the mice, but you do find by the neural network modeling. And you can go into these and find these in literature, and these are in fact well known, but the mouse studies never raised them. Okay, that's one example. In my last 5 minutes, I'm going to go through a second one, which will be out soon in August. This was done with Boehringer Ingelheim Pharmaceuticals, and it's on Crohn's disease, colitis, and Crohn's and colitis, inflammatory bowel diseases. And they're driven largely by inflammatory signaling, tumor necrosis factor alpha. So there's a lot of anti-tumor necrosis factor alpha therapeutics on the market. Infliximab, as Remicade, there's Humira and others. But like in cancer, these only work on a fraction of patients, and there's resistance in many. So Remicade, for instance, about 40% of the original people treated with Crohn's or colitis would be resistant to Remicade. And of those that it worked on, ultimately then another 70% would show up with resistance. So people are searching for resistance mechanisms. So you'd have a combination therapy, something to add with your Remicade. What else is going on that you also need to block? Well, people have tried it two ways. They've tried it with mice by themselves. So there's mouse models for inflammatory bowel diseases, and they might be tumor necrosis factor alpha overexpressors like this mice. They might be genetic mice that you have T-cell transplantation that then they react to microbiota antigens. So there's data sets from mice, both transcriptomic and proteomic, and then human clinical data tends to be transcriptomic. And you can look at differentially expressed genes between sensitive versus control, or resistant versus control, or resistance versus sensitive. Until now, people have been having a hard time finding really good resistance mechanisms by these methods individually. So we thought, why don't we put them together? Why don't we try to translate from the mouse models to the human models? Well, this is difficult for a number of reasons. Number one, the data are different. We have proteomic data in the mouse, transcriptomic data in the human. Secondly, the situation is different. In mouse, we have disease versus healthy. In human, we have resistant versus sensitive to treatment. Nonetheless, we gave it a try, and we, in fact, were successful. Here we used a different type of computational methods. We call it translatable components regression. It starts by constructing latent variables again and constructing them in the mouse space, just like before. So you take the mouse proteomic space and you construct latent variables there that represent the mouse biology. Then you take the human transcriptomics and project them into the mouse space. So essentially what you're doing is you're taking the human data and seeing where it falls in the mouse biology. Then what you do with the human data in the projected into the mouse biology, you now regress each of those latent variables against the human outcome of sensitive versus responsive. And what one's looking for is what are the principal components, or we call them translatable components now, that have the highest regression coefficients to the human pathology. And I want to warn you that it's not necessarily going to be the first 2 or 3 components, because those explain the mouse biology. Now we're trying to explain the human biology, so it may in fact be component 3 or 5 or 7. And you just have to, what you're finding is the human biology information is maybe in the noise of the mouse biology model. All right, it works pretty well. I'll just go through this quickly. What you do in the principal component space, you can find that the 2 most important predictors for the human responsiveness to treatment is principal component 1 and principal component 6. So as I warned you, it wasn't 2 or 3, it's 6. And what's shown here is how the different patients fall. Red are resistant, yellow are sensitive. And on principal component 1, you see what are the proteomics that fall there that are predictive of human resistance. And these are immune cell trafficking integrins, integrin beta 2, integrin alpha 4. This was already directly translated from the mouse, and it makes sense because it was on principal component 1. At the same time, on principal component 6, you find something that was brand new. Now you find other integrins and signaling, but these are signaling that happens on activated T cells through a different type of signaling activity, and this was not previously recognized for resistance to treatment, not found from mouse data. We've now validated that in follow-up experiments. I'll skip those experiments for the sake of time, but we've now in fact tested that inhibitors against this integrin signaling do in fact synergize with Remicade to shut down inflammatory signaling. All right, so what I've shown you is, I think, a new type of way to translate from animal models to human by humanizing computational systems models, and how you can find more biology that translates, and what are the key differences between mouse and human. With that, I'll thank the key people who worked on it. Doug Brubaker was a terrific postdoc. He's now started his own lab at Purdue. Elizabeth Proctor likewise, and is now at Penn State. Manu Kumar and Alina Starchenko are now out in industry. Collaborators, Kevin Haggis and Ken Lau, and then funding from the Army, Boehringer Ingelheim and NIH. And I hope that stimulated some interest. Thank you.
Thank you very much, Professor Lauffenburger, for this very exciting talk. And we have plenty of time for questions. So I would like to start with a question from Pablo Moreno. He asked, how stable is the performance of the mouse model when you change the way of deciding the bijection between mouse and human genes?
I would say some of these things we only know by what we've done so far. There's not too much theory on this. I think we're kind of breaking some new ground. So what we've done is a lot of cross-validation tests and permutation tests. We actually have changed different ways to project. We have changed normalizations. And so my point of view is always that I don't really believe something until we've done it multiple ways. And so it's not necessarily robust to everything you do. And so the key is to find enough results that do hold up when you've done them in multiple ways.
And Pablo continues with another short question. What orthology strategy you use, if any, to map a mouse gene to human gene to be able to use the mouse model on the human data?
Yeah, right now, that's we've been very careful about that to try to make sure we have good orthologs between a mouse and human, and we really only use data in which we have great confidence in that. You can imagine one thing I'd like to try is to figure out how we can do it when we don't necessarily have all the same molecular types, when some things don't necessarily translate. I don't really like imputation methods, but there is some, so the question is, what's not as good as an ortholog, yet you still have information about how it contributes to mouse biology and human biology. We haven't done that yet. That's kind of next on the agenda. So right now we're restricting ourselves to cases where we have very strong confidence that we got orthologs between the two.
Thank you very much. We have now a question from Justin. He asks, will the human updated classifier perform as poorly as a classifier constructed from just the mouse model if the mouse model itself is a poor model of the human disease?
Yeah, that's really interesting. So, I'm sorry, losing my voice a little bit here. So that's one thing that's very interesting about the second example I gave you. And that is, because remember in that example with the inflammatory bowel disease, the mouse model was healthy versus disease. The human data were all diseased, but treated versus not. So we didn't have any treated mice, or treated and then sensitive or not. And we didn't have any human controls. So you might argue there that those experiments, even though they tried to be a good mouse model for Crohn's or colitis, weren't really a good model because they weren't treated or we didn't have the right human healthy controls. And what happens, I think, is what you see there, that the data that translates, the information that translates, may be buried in some of the lower, I'll call them latent variables. They're more in the noise because they're not obvious predictors for the mouse. And because it's not a good model for the human. So one thing we're doing now, one of our next papers to go in will be kind of a reverse translation. We're working on Alzheimer's disease where there's a lot of different types of mouse models. And there's controversy over what the best human pathology characterization is. So we're working backwards. We say, okay, if we have some human cohorts with Alzheimer's, what are the best mouse models? And we deem what's the best mouse model by how strong a translatable computational model we can build. So that's my answer is the model may not be very good as the mouse directly, but if we can build a strong translation model, then it's a good model. But we just have to think about it differently. We can't think about it in terms of the observable. We have to think about it in terms of the information in the model.
Thank you very much. Now we have the next question from Begum, and he asks, do you have any entrance data and criteria for the data set you choose for model development?
Yeah, I can tell you we've worked on a number of problems, and often we find when we pull data from literature that there are problems. Well, I don't know. People look at things different way, and they normalize in different ways, and they compare in different ways. They have different noise thresholds. And once we go in and try to build the first animal model, we may be very unsatisfied with what the data look like, and we'll just stop there. Because if we don't feel confident in being able to construct the sort of machine learning model on the mouse data, then we're not going to bother to try to translate it to human. And that happens a lot.
Okay, thank you very much. The next question, well, actually the next from the audience is Salva, and she thanks you for the great talk. And she asked this question. For the second example, how would the overlap of the principal components from transcriptomics and proteomics compare to a partial least regression analysis of the two matrices?
Yeah, that's an excellent question. These all have been terrific. What a awesome group we have here. We've tried it both ways. I'm happier right now with doing it with the unsupervised mouse construction first and then regressing for the reason that was in one of the previous questions about the mouse phenotype not being the same. That if we construct the mouse latent variable space with a PLS, then I think we're biasing those exactly in a physiology that we don't necessarily want. So by allowing the mouse space to be unsupervised, we're just trying to find what's the biological structure, and then we don't have to undistort that to get the human. So you can do it both ways. I just worry that the PLS construction there, it just constrains you too much.
Thank you very much. Then we have a comment from Asa that she says, it's interesting to see that the mapping of transcriptomics from mouse to proteomics from human, that mapping sequence RNA protein that is usually tricky, even your human data. So thanks for the insight of your data. And we have the question from Delaram. And the question is the level of correlation between mouse and human gene expression related to how much similar the ortholog pair are?
I don't think so. There's some really great studies about comparing mouse and human gene expression. My MIT colleague Ernest Frankel, I think, is really one of the best at this. And he's shown that there's very poor correlation, mainly just because of the way the regulation is, the transcription factor levels, the epigenetic levels. It's not so much about the gene itself, that the differences are in the regulation, really at the transcriptional regulation level.
Okay, thank you very much. And I have a small question about the first study. You show that the comparison between the transcriptomic, the fold change between human and mouse dataset. You haven't mentioned the timing. Is the timing at all or at some point considered in this, or is just like a cells grown exponentially and then comparing the expression level without any specific phases?
Yeah, another fabulous question. That's that we're stuck with whatever was done in the studies. And that's a great point of contention. And so when I said there's a lot of controversy in the literature, that's one of the degrees of controversy, is to say, well, let's compare the timing and we got to throw these out because the timing wasn't right. I have a hard time knowing what the right timing would be, right? Because everything's different when you go from human to animal scales, and do you scale to onset of symptoms or things like that. So timing is a huge issue, and we just have to take it as given, honestly.
Would you know if there is a specific ways to, for example, to synchronize mouse cells and human cells in a specific way besides the serum, to synchronize it at the beginning of the cell cycle, for example, but some ways to synchronize and then to compare the level after synchronization?
Yeah, so the nice thing is, now that we've got some traction from published data sets, we now have new collaborations where people are designing experimental studies for this purpose. So we're working with some folks where they're trying to design this so they take data at multiple time points and they can argue biologically about which ones they think will be more informative. And our analysis is going to see if we can provide rationale for that. So,
Great.
So now going forward, we can actually build that into the studies.
Okay, thank you very much.